Good morning. Thanks one more time to come into our symposium. My name is Anderson and I have the pleasure to introduce you our next speaker, Dr. Yufeng Ge, who is an assistant professor in the Biological and System Engineer Department at the University of Nebraska-Lincoln. His research focuses in the area of advanced system as utilization of images technology for high throughput and plant phenotyping. He holds a PhD in biological and agricultural engineer from Texas A&M University. Please let's welcome Dr. Ge.
Hello, can you hear me? Well, first of all, thank you for inviting me to come here and give this talk. So the title I put there is a high throughput plant phenotyping green constant field, translational pipelines from gene discovery to crop improvement. Now, as I go along, you'll probably find out that I'm not going to talk so much of translational pipelines and gene discovery because that was the holy grail for plant phenotyping breeding. I'm trained as an agricultural engineer, so my specialty is really in sensing and automation for agriculture. So if I say something silly about plants, please be forgiving to me during the talk. Now, with that introduction, I'm gonna go through the talk here. First of all, I'm gonna give you a quick a survey in this talk about the things, the plant phenotyping things that University of Nebraska doing in general, and what I'm doing in my group in particular. And this picture is the high throughput plant phenotyping greenhouse, which is under operation now in University of Nebraska-Lincoln. And you can see these pictures, these nice pictures here. So that's what it looks like from the plant growing area. You can see all those plants and on the belt, and they are moving around at the time of imaging. And this is the imaging chamber. So we have a total of 4. So the plants go through them to take images usually daily. And then this is what it does look like during the night. So some of you probably already see this because this is they use this a lot in the promotional material for the university. So what I want to say is we got 5 imaging modules. So we have visible, and which usually we take measurement plant size, height, width, biomass accumulation, and growth rate. And we have a fluorescence imaging. So usually look at the chlorophyll content and the leaf senescence in particular. We have a near infrared, look at the water content. And we also have hyperspectral. So, and this one is the one that I'm going to talk about today. And I'm going to talk about sort of like a proof of concept study to look at its capability to measure water, micronutrients, and macronutrients in the plant leaf tissues. And then finally, we have a thermal infrared, which used for leaf temperature measurement. So the system got 672 pots, so it's fairly large in size. And we have 2 climate houses, so it can those environment is individually regulated. So if you want to do heat stress study, for example, actually you can do that. And then we have 3 automated watering stations as the plants go by. So it will automate to a very precise amount so that you can do the water study if you want to. Okay, so many of you, particularly when I talk to plant scientists, they are confused by the concept of hyperspectral imaging, even though the concept, people in remote sensing and engineering use that quite a bit. So it's basically it's an image cube. So it's three-dimensional, right? So you have a spatial domain, which is X and Y, and then you also have a spectral domain, which in the camera that we have here captures a total of 443 bands. So it extends from starting from 550 to 1750 nanometer. I'm sorry for the small font size there, but I make a little video here. So basically, just to let you see, how those different bands look like. I hope this would run. This would not run, which is unfortunate. But usually, what happens is, the plants would look quite differently along those bands. That gives you very useful information about what's going on with the plants, because of the chemical composition is different. The light interacts with the chemical composition differently, and then you can use that information to back infer what that chemical composition is. So, this proof of concept study is really trying to see if hyperspectral imaging can predict leaf water content and then micronutrients and macronutrients in the leaf. So we use the 60 maize plants and 60 soybean plants, and then so the experiment is easy. So we have water treatments. Basically, it's a controlled and then water limited experiment, and then we have a nutrients treatment as well. So we administer high, low, medium nutrients to the pots, and then as the plants grow to a certain growth stage, we feed them into the imaging chamber that you see before, and take the hyperspectral imaging. And after that, we basically destructively sample those plants. So we measure the leaf water content, and then we also measure the macronutrients, the micronutrients, to try to establish the correlation between them, right? So, and we do use partial least squares regressions. Thanks, Professor Gustav, to go through those statistics before, so that I don't have to spend much time here. So we basically calibrate the model with the PLSR, and we divided the whole plant population, 120 plants, into two groups. One is calibration, one is validation, and we build the model on the calibration, and then we test the model on the validation set. So the results that you see is all on the validation set. Okay, so well, this gives you a picture of how those plants look like under the RGB. So you can see on the top left is a maize plant under water stress, as opposed to on the right, it's a normal growing corn plant. So you can clearly see the difference, right? They're about the same development stage, and one is much smaller, and you can almost see the leaf rolling there. So it's quite interesting because you can see it, but it turns out to be quite challenging to develop a image processing algorithm to quantify the leaf rolling there. So there's people in our university trying to do that, actually. And then on the bottom there, so those are two soybean plants. So on the left is the nutrient deficient plant, and then on the right is a nutrient sufficient one. And you can also see the difference. So one is bushier and it looks darker green, so those are typical. And the one with nutrient deficiency is lighter in color, it's almost light green, yellowish, typical symptom for nutrient deficiency. So this summarizes the hyperspectral imaging process. So basically, we started with the cube, right? So we started with image cube, and then we extracted two bands out of the cube. So one is a 670 nanometer, which is the red band, and then we also extract the 770 nanometer, which is the NIR band. So we use those bands and we ratio them, and then we got NDVI. So NDVI is very sensitive to vegetation, basically, and then you can set a simple threshold to separate the plant pixel from the background. And then, so that gives us a binary image, so representing the plants here. And then we also introduced a third band, which is 1160 nanometer. And that one is useful because from here you can see the the difference between stem and the leaf, and we really utilize that bands to separate the leaf from the stem, so that we can actually just calculate the leaf, because stem isn't part of the measurement for destructive sampling. So after that, we use this yellow templates, right, to copy out, to go back and copy out all those bands, and then we average them as a average reflectance for that, representing that single plant, and then we use that to run the partial least squares against all those chemical data that we get before. And then this is the results of water content. So on the x-axis is the measured water content, and then on the y-axis is the image predicted water content. And so again, these are the 60 samples for the validation, not a part of the calibration purpose. So crosses, I believe, are soybean plants, and then circles are the maize plants. So you can see we got actually very good prediction, which isn't a surprise to me at all, because if you look at the remote sensing literature, the first thing they say is, those hyperspectral imaging can actually measure plant vegetation in vivo quite successfully. So you can also see a natural separation between those two species. So corn seems to have a little bit higher leaf water content for this particular experiment. And then let's move to the macronutrients. So again, we look at the nitrogen, phosphorus, potassium, and the sulfur, and all those are predicted pretty successfully. And we have an R-square close to greater than 0.8 for those properties. And then move to Mg, remind me what this is. This is magnesium. All right. And then we have calcium here. And those are predicted okay. So so we got R-square for 0.7. And so one thing here I can see is, when I look at these results, and first of all, I think this is very encouraging, because it seems like that you can do chemical sensing with hyperspectral imaging, and this is quite different from a lot of literature out there with high-throughput image-based phenotyping. Is they're all looking at other properties, like basically they are all doing structural physical traits, right? So they're measuring size, they're measuring growth, but this is actually another realm of phenotype that we can actually looking at. Magnesium and calcium are a little bit concerning, because it seems like the model actually not capturing the chemical itself, is actually capturing the separation between the two groups, the two species. So the crosses and the circles are not well mixed. So that's something that we certainly do not want to see in our PLSR model. And then come to the micronutrients. So micronutrients, some success there. So the results of the accuracy isn't as great as the macronutrients. So we can see, so zinc, copper, and then iron is all pretty encouraging here. And then sodium and the boron isn't really great. So this is certainly something that we wanted to do a larger scale later on, trying to sort out all those different things. Now I want you to keep in mind about the iron here. So it's got R-square of 0.68, because the next one that I'm going to introduce is the field phenotyping work that we do for the iron chlorosis deficiency. So I just put a note here. Let's see. So a quick conclusion here. So hyperspectral imaging is useful to quantify a number of plant leaf chemical properties rapidly and non-destructively. So we see that leaf water and the nitrogen the best, and the sodium and the boron poorest, and other thing between the sodium is the poorest is a little bit discouraging, because I know that there's a lot of people doing salinity tolerance, right? So if you can actually measure sodium non-destructively through the leaf tissue, then probably that can be a great tool for for salinity study. So, and then we certainly have limitations, right? Again, because this is just a proof of concept thing. So we wanted to involve more crop species, and we wanted to look at more, within a species, we wanted to look at more genotypes and more developmental stages, because we figure that all those probably would play a role in your model building process. So that's the first one, that's in the greenhouse. And then, so the second one I wanted to introduce is the field phenotyping effort that we have. So this phenotyping card is what I built since I start in UNL. So structurally it's quite simple. So 2 bicycles put side by side, right? So I just want to start something quick and being able to, start to collect the field phenotyping data. So, but if you look at the sensor side, it's actually quite serious. So we have 5 different sensor modules on it. So we have, so you can see this is actually a webcam, so that, which allows us to take RGB camera. And then this is the height sensor. And then that's the NDVI sensor. And then we have, this is the fiber optic. And then so the fiber optics go and connect to a compact portable spectrometer on the platform. And then this is the canopy temperature sensor. So these are basically the canopy, crop canopy sensors. And then we also have other environmental sensors on it, so which those are on the top here. So we have air temperature, relative humidity, and then we have PAR intensity. So PAR is a photosynthetic active radiation. And then we also have GPS signals. So so basically the product of this sort of search, so every time that you go out for survey, and the product would be basically a map that allows you to see all those indices on the spatial distribution of those indices. So we have been quite successful. I'm glad that I was able to effectively plug into the breeding program there. So some of you may know Steve Benziger, some of you may know George Graff and Daniel Sketchman. Those are all, and James Schnabel, they are also. So I was able to collaborate with them and test the phenotyping system across the state of Nebraska. So we have a couple of crops scanned already. So in the past 2 years, all these red symbols are the areas that we have done already. And then, this year we're going to add another crop account. It's a camelina that we got funding from USDA and NIFA to do this work, which is very appreciative. And so that's in Scottsbluff. It's very dry, arid area. So this is so the idea is we are able to move this thing around and we can do cross the environment. So the one that I wanted to talk a little bit more in detail is soybean iron deficiency chlorosis. Again, this is the collaboration with Dr. George Graff there, the soybean breeder in Nebraska. So he introduced me to this research phenomena, and so it's basically an abiotic stress, right? Exhibits stunted growth and distinct yellowing of leaves of interminal region. A lot of those doesn't really make so much sense for me, but I'll just read it. And so the symptom, as I understand, show up easily in calcareous soil of high pH and free carbonates. And then it reduces yield and costs producers a lot of dollars, right? And then it's quite a complex problem. And it's a very active research area, and they want to fix the problem and understand it from the genetic level. And then it's the best time to phenotype is really at the V3 before stage. So what we do is, so he's preparing all the genetic material, and he has a trial out in this location, Valley, Nebraska. And then we test the 64 genotypes with the 4 replicates, so about 250 plots there. And then, so we bring our field phenotyping system and we push through the the field and collect field images. Those are native view images covering the whole 2-row plots. And this is done at the V4, V5 stage, so it's a little bit off the optimal time, because the V3 really can't get there because of the weather, if I remember correctly. So then we do image processing. Now, what I want to emphasize is the objective is to test the 3 sets of scores. So the first score is really the field score. So that's how the breeder do the scoring, right? So you have a pencil, you have a pad, and you go through the field and you write down the scores. So you score them 1 to 10, 1 to 9 basically. And then so the second score is, since we have the phenotyping, we collect all the images. So what we do is we have the same graduate student who do the field score, and now we will have them sitting in front of the computer and look at all those images, and he do the score that way. So basically, it's you look at the digital image and we do the score. So we call that an office score basically. And then the third step is we remove him completely out of the picture. So basically, we develop image processing and we do an automated score. So we let the computer to figure out what it is, and then we wanted to compare those 3 sets of scores. And then year data later became available to us, so that we can also run a correlation between yield and the 3 sets of score, which is always nice to have. So this gives you an idea of the IDC scores from 1 through 9. So 1 being the healthiest, and then 9, if you move to 9, it's pretty dead, so nothing's there. So this is the picture from our phenotyping system. So I wanted to just say a few words about image processing that we are doing. So we start with the image here, and the first thing is you extract basically the plant, what we call the plant pixels. Right? So remove all the soil backgrounds and dead plant debris, and those are all out, and there's only plant pixels in there. So and then from the plant pixel, we divided them up into 3 fractions. So so we basically convert this RGB mode to what we call the HSV mode, which is a different color mode, but tells you a little bit more about the color of those pixels. So we use that color mode. We divide them into 3 fractions. So we call it a green fraction, and we call it a yellow fraction, and we call it a brown fraction. So the idea is really it's IDC when the graduate student out, he does the field scoring field. It's really he's trying to see if the leaf is turning yellow or brownish, and he's if that happens, so he's scoring that plot a little bit higher in the scale. So so we are trying to see, well, if this the yellow fraction or brown fraction can actually be a proxy for the severeness of IDC in this automatic scoring processes. So so that's that. And this slide compares 3 sets of scores. So I put the field score on the x-axis, and then so the office score is so this plot is on the y-axis, and you can see the correlation between them is 0.67, and you can see that they are not consistent, and they are usually the office score is probably 1 or 2 score higher than what they do in the field. So this is the same person, so it should be. We're still trying to figure out what makes that difference, right? So you can see a lot of those points here, meaning that they are off by 1 or 2. And then if you look at the comparison between field score and the automated computer score, and they are all the distribution looks like this. So one of the difference between algorithm score is the algorithm score is really continuous, so it doesn't like a human. It says call 1, 2, 3, right? So you can have a continuous distribution, and this is the reason why you see all those different the distribution of points there, and they are all the correlation are all quite significant. So they're not bad, even though they are inconsistent to each other. Now, if you look at, well, so this is a slide, I'm showing the slides there because I'm giving instrumentation class to our undergrad students. And so I like this because, this slides clearly show that our interpretation of color, brightness, and the size motion isn't scientific, is an objective way. So it's a psychological and physiological process. Right? So if you look at the top one here, now if you focus your eye on this cross here, then you will see a green dot revolving around. So that green dot isn't really there. It's because it's a complementary color, and your brain create that for you, right? So it's not objective. And then this is another very good example, relevant to breeding and phenotyping is, the square A and B, they're actually the same brightness, but because of the shade that you have, so B is perceived a little bit brighter than A, right? So you can see this, they are the same. But so this has been used a lot in the engineering as an example, but I feel like, plant breeder probably don't see much of this stuff, right? So so you can see how this can apply to the field of plant breeding. Is you have people go out and look at things, and if you have different graduate students, because of their own psychological, physiological processes, their interpretation of their score is going to be different. Let alone that if you have a sunny day, if you have a winter day, a cloudy day, or if you do this in Nebraska or in Kansas, it's Iowa, I should say. Sorry, it's all different, right? So so that's why it becomes challenging, and this is why I think the automated high throughput phenotyping would play a role in here, is because you really bring that objectivity into here using sensing, sensors, and instrumentation. Okay, so and again, these two guys, they are the same size, but this guy's interpreted a little bit larger, right, because of the background in there. So. Okay, so that's that. And so now finally compare 3 sets of IDC scores to yield. So this one is the yield versus IDC field score. And we got a negative correlation, which should be negative, of 0.43, right? So you can see a line here. Now if we move to the office score, you can see office scores actually have a higher correlation absolute value, which is very encouraging, because this means that what you do in office is probably better than in the field, right? And I haven't really tested if this difference are significant or not, but we just for now we just look at the magnitude, and then to move to the yield versus IDC automated algorithm score. So this value is quite similar to the office score there. So results so far is quite encouraging to me. So we're going to, and this is done in 2016, and we are trying to do another round of experiment this coming season to see if we can actually validate or see this pattern again. So I throw in this here, and I think this is quite interesting. I hope this would play. So this is the phenotyping system that we developed, right? So we push them through in the field manually, and it turns out it is really it's very tedious, and you have to have 2 people to do it. So one of my postdoc, he says, well, can we automate it? So what he takes initiative and try to do it. I mean, this is really, to me, it's a little bit ridiculous, because, in agricultural machinery, automatic navigation, people do it all the time. If you go to John Deere, they will say, well, we already done it, right? But in plant breeding, nobody really trying to invest time and money to develop this kind of technology. So so it takes one of my visiting scientists a year to develop a system like this. So right now it's all GPS guided. We're trying to look at the GPS position, and we have the waypoints in the field, and they're trying to see if we can correct the routes through all those points. We still have some glitches, but the system is already there. So this is if breeding can create an industry to do this, then I think John Deere people would, if they see a profit, they're gonna come, right? So yeah. Okay. So next, so I'm also going to kind of say 3 other projects. Is again very engineering focused project that I have. These just started last year, so I don't have a lot of quantitative results yet. Just kind of give you a quick overview of the breadth of the project that we're working on. So this one is the phenotyping robots. This is supported by NIFA. So I borrowed this picture from a colleague of mine at Purdue University. So this really is the future vision of the greenhouse, right? So we have UAV flying there, and I don't know what this is probably MRI imaging system. And then we have all those robots that are taking, permanent, consistent care of the plants, measures them like 24/7. So this is the phenotyping robot is really trying to address that part of the greenhouse. And so this is my, we see this all the time. So this is, I believe, this is the photosynthesis measurement system, and that's the stomatoconductance measurement system. But my graduate student has to, every time he measure plants, he has to do this, right? So our idea is, well, can we, you can see the common points between them is you need a finger, and you need 2 fingers, and you need to stick the sensor to clamp them on the leaf. So can we develop something like this and ask the robot to do this for us, right? So this is the whole idea of this phenotyping robot. Now you can launch many of these phenotyping robots in the greenhouse so that you can eliminate you can have the graduate student to do more valuable things in the lab, right? I mean, in the computers to do the data modeling, right? Interpretation would be more important than this if this can be automated. So this is what we are trying to do here. And so I just showing 2 pictures. So that's the real thing. The one that you see before is a model, and this is the special gripper that we developed. It's actually can measure the reflectance of the leaf, and we are almost there. There's a lot of engineering challenges there. So how do you kind of recognize the leaf, and how can you rank the leaf actually? Because there's certain if you look at the corn plants, so, there's a certain leaf that you want to measure physiologically. Reasonable, right? So he has to do, if there's 3 leaves detected, he should be able to kind of rank the priority, like which one go first, if this fails, the second one, we go to the second one. I don't think this will play, so I'm gonna, it won't, but I'm gonna just kind of, so this is the video here, so you can just kind of let you see what it currently stands, and it's not very exciting, but it goes and it stops there, and it clamps on a leaf. So all these it goes through the image processing procedure. So that's that. Let's go back to slides. Okay. All right. So I hope this is going well, and I hope we can have good results. If I can still come next year, hopefully I will have quantitative results to show you, how effective the robot is, right? How does that compare to a graduate student, basically? Okay, so the third one is a multispectral 3D laser phenotyping. So this work is actually supported by NSF, and so our idea is, if you look at the pictures, the plants is the real 3D structure, and you basically you project that to a 2D plane, and we're trying to inverse that 3D information from the 2D structure, right? 2D image, I mean. So our idea is, well, can you just use laser to do the scanning so that you can scan from the side, so that you can get the 3D point clouds and be able to do better job to look at the leaf area or leaf orientation. Because theoretically you can actually get leaf orientation if you can reconstruct that surface from the point clouds. And so this is the setup in the lab, and we intentionally get a ornamental plants there. And this is the LiDAR system commercially available. And we just right now at this stage we just wanted to get the point clouds so that our computer scientists can continue their work. But our idea would be to develop a multispectral 3D laser. Now there are a few advantages with the laser. So first of all, it's very focused. And it can inject a very small, very large. The energy density can be much higher, right? So that we can potentially get laser-induced fluorescence of the plants. And if you are trying to do PRI, those vegetation indices, they all require your bandwidth is very narrow. So laser can really meet that requirement. So the multispectral laser is really originated from those thoughts. And what else? So this is the video, and I don't think this is gonna play, and I will show you here. And again, not very exciting for now. And this is so the laser actually is scanning now because but that's in the infrared laser, so it's at 900 nanometer, so you don't see. So it would be much better if I throw in a green laser, right, so that you can actually see the scan. The laser goes up and down, and then the rotational plates rotates 330 degree, 60 degree. I mean, so that you can actually see an effect. So, but we do have some results here. So this is okay. So that's what does it look like for now. So pictures, right, and then that's the 3D structure. And we still have a lot of noise, and I'm a little bit headache on how to process them now, but the idea really is trying to reconstruct the surface from these points, right? And once we can describe that surface in the 3D functionally, then you can do all kinds of cool things. Right? You can derive the leaf area more accurately, and you can see the angular distribution, because the surface has already been reconstructed, and you can do that. So, but I know that we're not there yet. So I'm counting on my collaborator in computer science to do that for me. So that's that. And finally, this is the big thing now at University of Nebraska-Lincoln. So we are developing a field phenotyping facility. So the scale is enormous. So this is the plant growing area. So we have 4 poles, right? So there, and it's basically a cable robot idea. So that you have like 200 plots on the ground, and cable robots can precisely control where that needs to go, and then go there and take images. And you can the good thing about cable robot is, compared to the ground-based system is, with the ground-based system, if it's wet then you are done. You can't go there anymore. So, but this is fairly good, and we tested. So Nebraska is pretty windy, but this cable robot is very stable. So because of this special design, you can see it's not 4 cable, it's 8 cables, so that it's really pin it down to hold it onto the position. And this is what it looks like for the dolly. So that's the instrumentation dolly, and we will have a computer up here, which is protected by the ray, and then we'll have all the sensors and instrumentations down here. So that's so right now we are having a thermal camera, we are having a LiDAR, we are also having a spectral camera, and we'll have the spectrometer, so to look at the reflectance. So this is what it currently stands. So you can see 4 poles, and I believe they are installing this thing now. So they are installing the dolly and the winch, what they call the winch, to control the cables. So this is, so my group there is really to develop an instrumentation package. So we are going to, so we are responsible for this pen tilt unit, right? So, and we'll have all kinds of instruments too there. And this is really exciting because I feel so lucky that, you know, we're able to plug into the overall efforts there, being able to do this kind of for research. So the plan is for us to be operative by June maybe. So we are very hopeful that we can get some data this year. And you know, if you happen to visit Lincoln, give me a call, and I will show you this this outstanding facility there. So okay, so the last thing I want to say about this field phenotyping facility is, so this is quite unique. So what you don't see above ground, but they are underground, is the subsurface drip irrigation. Okay, so this entire field is divided into this like 130 little plot area, and then you can see all the control valves, meaning that you can actually administer a precise amount of water to each of those little cells. So this is really a I should say, a semi-controlled field environment, particularly for water application. So, so if you want to have a drought study, you know, if you wanted to look at drought resistance, you know, this is a very good facility to do this kind of study. Okay, so I guess that's it. So I wanted to acknowledge the funding agencies, really, so without them, nothing is possible. So we have NSF and USDA Night Fund, and we also received funding from North Central Sun Grant, and the commodity board from Nebraska has been very supportive of our work here. And with that, and postdoc and graduate students. So I can't put all their names here, but they are really the driving force for the research everywhere. And I want to acknowledge them as well. And our collaborators. So I found myself in a very awkward position sometimes. You know, I have meetings with a group of plant scientists, and I sitting there, and they talk about things like I feel like sometimes I feel like a moron, like because I really don't understand what they're talking about. And I'm, you know, coming from a very different perspective, engineering background, but I'm very grateful that I have the opportunity, and it has been. The process is really rewarding, very, very, very rewarding for me. So with that, I would like to close it up. And if you have any questions, and I will try my best to answer them, okay?
So my question is, in your first study, we were looking at hyperspectral, which is testing nutrients. You said it was poor for sodium and boron. Would a different band work more effectively for certain nutrients? Did you investigate that?
So, so we using PLS. So it is a data-driven method. So basically, PLS look at all wavelengths and then come up with the best combination to do the prediction. And I think the problem isn't so much of can the hyperspectral predict boron or sodium? I think people at UNL also reminded me about it. Is maybe our experiment isn't really the greatest, because we do not isolate the boron or sodium from all other nutrients. So when we say we administer the nutrient deficiency to the experiment, we basically just have different levels of nutrients, include everything. So maybe there's some sort of like a compaction issue. I don't know, but I feel like that if we wanted to say for sure that whether hyperspectral imaging can or cannot predict sodium or boron. We should make a more careful study to keep all others constant, right? And then vary the sodium or boron to see if it can actually predict. Now, it's the limitation really is it's probably quite expensive to do so, unless that you can write a very convincing proposal to NSF, say we're going to do this, then you have the money and you can do it. And otherwise, for this kind of a study, I'm very reluctant to say I wanted to keep everything else consistent, trying to just vary the sodium in the plants. I hope that answers your question.
My question is, you have the images, and based on these images, you can generate different trees. For the water content or nutrients, you can generate different tree data. And in the image processing, what is, for the different data, what is fixed, what is different? I mean, the precise, maybe for different trees, the bands is different, or the wavelength is different. And even for the same tree, for the different species, what is you should adjust or what is fixed. So it's how hard we can follow this processing.
Well, I'm not sure I understand it, but I'm trying to answer this to the best I can. So you're asking about the traits. So all the image traits are indirect, right? And this is really one great thing about image is, you can measure anything, pretty much anything you want. You can look at the size, you can look at the width, height, and you can look at the convex hull, you can look at like the bushiness, so basically how bushy it is. But those are not the conventional traits we are using yardstick to measure. So those are based on images, right? So and they are advantages for it because you have the image and there you can have automated routines to figure out all those images. And what happens is you just like genotypes, and you can have thousands, literally thousands of even phenotypes to measure. But the challenge is, first of all, are those phenotypes all necessary, right? And the second of all, if you find some significant phenotypes, let's say, is that really carry any genetic meaning, or is it just like a false or spurious discovery because you got so many variables? Something ought to come up because you set a threshold to 0.05. So this is really something I think we should do. Is we need to be ambitious, and we also need to be cautious about image-based phenotyping. It's a little bit different from a traditional way of doing phenotypes. Is you can measure so many of them.
I have a very simple second question. So you have the environment sensors, and you measure different environments. So when you generate this data, do you adjust use this environment?
That's a good question. So the question is, we measure the environment data. Do we use that? Yes. The answer is yes. For example, one variable we use is really air temperature, because as we go along, so let's say we started at 9 a.m. and usually we started at 10 a.m. and we're trying to finish up everything before 2 p.m. because that's what the literature say that's the window that you should really start to do the phenotype measurement. But even with that time window, as we go across through the field, the environment changes, right? So particularly like air temperature, it will change actually pretty substantially during that window. And so one thing we do is we measure the crop temperature and we adjust that with the air temperature. And so it's a TC minus TA. And that's the TC is the canopy, and TA is the air temperature. And that is really a better way to probe if crop is stressed or not. So yes, we use those information.
Yeah, that was related to my question. How long for the field-based phenotype, and I understand it's still under construction, but how long does it take to get across the entire field and collect data from all the plots?
You mean the cable robot?
Yeah, the cable.
Yes, so we really don't know yet. So it moves pretty fast. I hope next time when I'm here, I can have a video to show how it goes through the field. But I think it's fast. So move from one corner of the field to the other corner. I think it'll take like less than 1 minute.
Okay, but then deflect data.
Right. So the limitation is really how fast we can collect the data, and I cannot answer that question yet. So my best bet is we should be able to collect one plot within 20 seconds. So 10 or 20 seconds. So don't know yet.
Thanks, Rico. And I'm interested in the IDC work since we're in the similar work here. And I must say for the IDC, when you use the image, automatically wrapping the images, do you use something other way to training model, and how do you create this the automatic score? Right,
that's a good question. So what we do is we do the same. So we have divided the images into two sets. So we have a training set, we have a validation set. So what you see, everything there is a validation set. So we do train the model so that, I mean, I think essentially we don't really need a score from 1 to 9. We can just ask the image to output some number, which is consistent to the IDC score that you would make, and then we can do the comparison that way. And it's the same, right? So you can score everything 1 to 9, which is we do here, but it's not necessarily the case. I hope that answers your question.
Thanks for your talk. Mine is related to his as well. So you do 3 different sort of phenotyping sets: the office, the field, and the automated. Would in this case 3 or 2 better than 1, integrating the different scores to help improve whichever you prefer? You seem not to like spread students a lot.
Yes, I mean, this work is really trying to figure out an actionable way to do, to address some real problem, right? Anything that can alleviate the effort that the graduate student needs to spend in the field, we're gonna do it. And regarding trying to merge 2 sets or 3 sets of data, that's certainly a possibility. Is that what you're asking?
Yes. So have you done that? Do you think the scores that a grad student would help maybe improve what the automated score would do? We give you a better correlation between the automated score and the yield that you're trying to do if you use whatever scores are made in the field to train your automated score.
Right, yeah. So this is what we do. We use the field score to train our automatic so that it actually gets into the scale of 1 to 9. But as I said, you really don't need to do that. You can just get the number from whatever the computer throws out to you, and you make an assumption that those numbers, even though they are not from 1 to 9, but it's still somehow correlated to an IDC, then you can use that directly.
Hi, thank you for the great talk. My question is pretty general. Where are you putting all this data? Where are you storing all these images? We're talking terabytes. What do you do with them?
So the question is about the data curation and data storage, and this is it's a contentious topic. People are talking all the time. Now, for our greenhouse data, we do have a dedicated database that we have. We do the calculation for the full-time operations of the greenhouse that I showed. The phenotyping greenhouse create about 10 terabytes of data every year. So it's not really that big. It's 10 terabytes, but still it's a significant amount of information. But we do have a database that is there, and all the researcher, right now it's still a little bit glitch between them, but the weekend they do have a web-based interface, if you will, that they can download the data from the database. And I'm not sure what is the the license issue or the copyright issue. I'm not really sure. So right now, I think what I see is the PI would be responsible for their own data. So if the experiment belongs to you, then you have to say on, do I want to share this data with other PI or do I not? For the field phenotyping data, no, everything is still on my computer. It's not going anywhere. But I see this as really an important issue. If I already see people are interested in IDC, if you are adopting the same technology here at Iowa, and there ought to be some way to compare the data. There ought to be some way to do this in a fairly consistent way. So in that regard, I think a commonly shared database would be really important to do it rightly or even stimulate other new discoveries. So I hope that's my 2 cents. I don't really have a good answer.
We have a webinar running, and one of the